So welcome to week 6 of the lymphatic system. Most of the lymphatic system is, I think, pretty intuitive until you get to immunology. We're going to talk about functions of the lymphatic system, lymph and lymph vessels, which are going to look really familiar compared to our, let's say, veins, for example. Then the lymphatic cells, and they have a relatively complex cascade that we're simplifying, and lymphatic structures, so there is a bit of anatomy associated with these. We talked about in our cardiovascular system how we're going to lose fluid into capillary beds because of hydrostatic pressure, and we're going to get some of that fluid back via osmotic pressure of the capillary bed. But we also recognized last week that we're going to have some more fluid leaving than actually comes back. And that's where the lymphatic system comes in. We're going to add in that there's more lymphatic vessels that are going to pick up the excess interstitial fluid and ultimately return it to the cardiovascular system. And on the way, they're going to filter it through some lymph nodes, and they're going to look for invaders, pathogens, and that's going to help our immune system. So we have to talk about both the immune system and the anatomy of the vessels, but the vessels themselves are pretty intuitive. One thing to understand is that it's all the same fluid. You consume water through your GI system, and gets absorbed through your GI into your cardiovascular system. And at that point, we call that fluid plasma. The fluid part of your blood is called plasma, right? Then it leaves your blood vessels through the capillary beds, and then that same fluid we will now call interstitial fluid. And then when it gets picked up by lymphatic vessels, it's still the same fluid. It's now called lymphatic fluid. So the composition may change very slightly. For example, you've got more plasma proteins and obviously formed elements and cells inside of the bloodstream, whereas you should not have very much albumin out in the interstitial fluid or in the lymphatic fluid. And additionally, lymphatic fluid may have a couple of other constituents, but generally it's the same fluid. We just give it a different name depending on where we find it. Anatomically, lymphatic vessels are unique because there's no heart pump associated with them. They start in capillary beds. They start as these little dead ends. So they have a sort of capillary-sized dead end that will be distributed throughout capillary beds. And they'll be made of simple squamous epithelia. And you're going to have at this very, very microscopic layer these physiological valves made by these overlapping single squamous cells. So as interstitial fluid is found in this interstitium, it's going to be easy for that fluid to go in, but once this lymphatic vessel is filled, it will compress the edge of the cell such that it will shut that valve. So even at the microscopic layer, you have physiological valves. As we get into larger lymphatic vessels, we'll see actual valves derived from endothelium, very much like you would find in a vein. So they do have valves. This is all going to be one way. We start in the capillary bed, and we get larger and larger and larger, and then we return blood to the cardiovascular system. So it's all a one-way system, not like the cardiovascular system where it's going out and it's getting smaller and it's coming back and it's getting larger, right? It's one way. So again, lymphatic capillaries, closed-ended tubes. They have a dead end. Fluid is pushed in and doesn't make it back out. Overlapping cells make one-way valves. There are going to be some specialized lymphatic capillaries. We're going to find them in the digestive system. And again, we do have respiratory and digestive coming up right after this, so we're just kind of segueing really smoothly into those topics. In the GI tract, you're going to have a lymphatic capillary lining the walls, or within the walls of the small intestine, and those are going to be called lacteals. They're actually going to have the job of absorbing fat from the things that we eat and carrying that to the liver. So that's going to become more important as we get into the digestive system. So interstitial fluid as well as lipids and lipid-soluble vitamin. Lipids are fats, fatty acids. And that fatty substance that you pick up from the GI tract, we're going to call that chyle, C-H-Y-L-E, chyle. Not a person named Kyle, it's just called chyle. So we get into slightly larger lymphatic vessels, and now we do have specially derived valves, not just physiological valves from overlapping cells. Still, it's going to be one-way flow. Here it is histologically. You've got a simple squamous epithelium still with this lymphatic vessel. This whole thing is a lymphatic vessel, and you can see the valves here. At this point, it may be worthwhile to note it has taken us forever to figure out the lymphatic system. This is actually a little bit more modern. A lot of our research into the lymphatic system has been really limited until very, very recently. And histology is a part of that, because if you're looking at this simple squamous epithelium, it can be difficult to differentiate things like lymphatic vessels versus veins at some levels. And even then, especially at the macroscopic level, you can't really see lymphatic vessels except for a couple of really, really gigantic ones that we'll get to in a second. So a lot of this was really mysterious. A lot of this is guesswork. We actually didn't find the lymphatic drainage of the head until a couple of years ago. Like literally 2-3 years ago is when we figured out that we actually do drain lymphatic fluid from our heads. That's how long that took, yes.
So are they just smaller than the granules?
They're very small, yes. They're small, they're going to compress very easily. In tissue, this is going to be something that's going to be hard to find histologically. We knew there was another system of tubes. We knew there was a series of tubes out there somewhere because we saw the big version of those tubes, and you have this really cool anatomical feature called the cisterna chyli. We just didn't know what it was coming from. Like, what is this? So again, lymphatic vessels get larger. For certain structures, since this is a one-way system, when we get to lymph nodes, we're going to have afferent and efferent lymphatic vessels compared to those lymph nodes. And you've already seen this naming convention. What does afferent mean? Arrive. Well, I use A for arrive, and then E for exit. So A is going to be arriving at a lymphatic vessel, and E is going to be exiting a lymphatic vessel. So to a lymphatic vessel, from a lymphatic vessel. And again, it's going to be from the, or to the lymphatic vessel, arriving at the lymphatic vessel from capillary beds, and efferent lymphatic vessels are going to take away from the lymph node and head back to the cardiovascular system. So again, this is all one-way stuff.
What is that similar to?
Afferent and efferent is a neuroscience term that you see a lot. So efferent pathways, afferent pathways. We'll see it again in the renal system. We're going to see an afferent and efferent arteriole to a capillary bed. Okay, here are our really large-scale lymphatic trunks and ducts. Now this is an asymmetrical system. You can't rely on the naming conventions being symmetrical in this case, and you'll see why in a second. It's not going to make a ton of sense. So way back in the day, before we really had a good sense of the lymphatic system, we just realized some people had this big old pouch right here called the cisterna chyli. And that was coming up with this big vessel. It's not really an artery or a vein, but it's called the thoracic duct, and that drains right to that left subclavian, and only the left subclavian, not the right subclavian, the left subclavian. And we were like, well, that's weird. And if you want to be kind of disturbed for a second, I can tell you a little bit of a story of how we got a little bit more in depth with the anatomy on these. I might even pause it because it might be kind of gross for some people. Okay. So again, the big structure is a little bit oriented to the left, and it's the thoracic duct. Now you do have a slightly smaller duct on the right side, but it's going to be known as, make sure I get this right, right lymphatic duct. So usually I say when there's a right, there's a left. That's not true here. There's no left lymphatic duct the way there's a right lymphatic duct. There's a right lymphatic duct, and there's a thoracic duct. As for trunks, you will absolutely be held to what region is drained by which trunk, I do think they're intuitive. Just because they're intuitive doesn't mean you don't need to study them. Jugular trunks are going to go in parallel with that jugular vein, which makes sense that they would drain the head and the neck. Subclavian trunks are going to drain the upper limbs, breasts, and superficial thoracic wall. Bronchiomediastinal trunks, if you've got your terminology down, you know that this is going to be your bronchi, your mediastinum. That's going to be deep thoracic structures. Your intestinal trunks are going to do most abdominal structures, your abdominal viscera. And your lumbar trunks are going to go from your lower limbs and your abdominal pelvic wall and pelvic organs. So I will ask you to illustrate this today. Do a color coding if you can. Sort of, stick person outline. I provide you with the outline. I just want you to color in where everything's draining from. Obviously, when you're talking about superficial versus deep thoracic, you got to be a little creative with how you illustrate that. But this is what I'm asking you for on your worksheet today. Now, one reason that you have a big old thoracic duct and a right lymphatic duct, which is smaller on the right, is that lymphatic drainage is not symmetrical. You actually have 3/4 of your body draining through that thoracic duct, and the remaining quarter is drained by the right lymphatic duct. So it's the border is that respiratory diaphragm and that mid-sagittal line. So from your right respiratory diaphragm and your mid-sagittal line upward, this is drained by your right thoracic duct. The rest of it, right lymphatic duct. See how dangerous that naming convention is? I mess it up all the time. The rest of it is through the thoracic duct. Everything else, all the rest of it. So again, that thoracic duct does drain to the left subclavian. The right lymphatic does drain to the right subclavian and at that junction of where it meets the internal jugular. That super cool thing, that big old pouch we found way back in the day. Not everybody has a pronounced cisterna chyli. It's not obvious on everybody. There's a lot of individual variability. And it collects lymph from pretty much everywhere, that whole 3/4 that we showed earlier is going to pass through the cisterna chyli and back up through that thoracic duct to the left subclavian. Questions so far? Yes?
When you were going through the trunks and the jugular trunks and whatnot, on the page before, there's like a right jugular trunk, but there's not a left. Is that part of the...
I'm sure there's a left jugular trunk.
Okay.
It's probably just not illustrated there. That's a good question. Since this is so asymmetrical, right, there would be a possibility that the rest of these trunks would be asymmetrical. I believe, for our purposes at least, these are going to be symmetrical. We'll have the trunks be left and right bilateral. I do like this dissection. You can see they have shown this thoracic duct as it drains into that subclavian right there. You can see that it is a rather large structure. Otherwise, the only structures that you can see for the lymphatic system are lymph nodes on dissection, the thoracic duct. You can see the right lymphatic duct and the cisterna chyli, and that's kind of it. You can't see any individual lymphatic vessels on gross dissection, which is cool and weird, in my opinion. Okay, lymphatic cells. We've already seen some cells big picture. We've already seen our never let monkeys eat bananas, our neutrophils, our lymphocytes, our eosinophils, our monocytes, our basophils. I did those out of order. I switched 2 of them, rather. We're going to get a lot more specific today. Mostly we're going to zoom in on the lymphocytes. You do have a couple of specifics here. Nurse cells. I don't think you're going to find it otherwise in this PowerPoint, but it may be a lab practical type question. They're going to help with the development of some of your white blood cells. Dendritic cells. Actually, I have to kind of look that up. Sorry, guys. You're going to have regulatory cells that help shut things off. Mostly we're going to focus on these guys, T-lymphocytes, B-lymphocytes, and natural killer cells. And yes, natural killer cells are as cool as they sound. In fact, they're cooler than they sound. They are awesome. We like natural killers. Now, it's not just 3. You're going to have subtypes of T and B lymphocytes. So I'm going to go through this cascade once, and I guarantee by the end of the day we're going to go through this cascade about 500 times. And that's going to be how long it takes to understand this cascade. I'm only going to record it once, though. We're going to start with the helper T lymphocytes. The helper T lymphocytes express a protein called CD4. The ability of the helper T lymphocytes is that they release cytokines. Cytokines are chemical signals that are going to attract other elements of the immune system. So when a helper T lymphocyte recognizes that there's a pathogen, it's going to release cytokines. And those cytokines are going to, I'm air quoting, call some friends of ours memory B lymphocytes, cytotoxic T lymphocytes, and macrophages. And what were macrophages? You already know this one. Big eaters. So they're going to bring the big eater because maybe it can consume the pathogen. And then we're going to talk about what those B lymphocytes and T lymphocytes are also going to do. So again, this was a helper T lymphocyte. This is a cytotoxic T lymphocyte. So it's actually calling another type of T lymphocyte. The second type of T lymphocyte is the cytotoxic T lymphocyte. And it expresses the protein CD8. It was activated by cytokines from the helper T cell. What the cytotoxic T lymphocyte actually does, it's capable of inserting a perforin molecule. It makes a little tunnel in the cell wall or plasma membrane of the pathogen, and that tunnel being there is going to lyse it. It's going to lose all of its water, and that pathogen is going to dehydrate to death, which is kind of cool. If you're thinking about it really hard, like, that's awesome, right? Anybody? Anyone is thinking that's really weird and cool? Okay, so that's fine.
So it was perforin? How do you spell that?
Perforin. P-E-R-F-O-R-I-N. As in perforate. And so it's kind of in the name, right? Cytotoxic T lymphocyte. It's toxic to cells. So if that invader, that pathogen, has a plasma membrane to insert it into, then it can lyse it. And that's pretty cool. The other cell that was invited by the cytokines, called by the cytokines, attracted by the cytokines, was the memory B lymphocyte. This is the memory B lymphocyte. What the memory B lymphocyte is going to do, it's going to undergo mitosis. And that mitosis is going to produce more memory B lymphocytes to carry on the memory of this pathogen. But it's also going to differentiate into plasma cells. So plasma cells are another type of B lymphocyte. And the job of plasma cells is to secrete antibodies. So you've seen antibodies before. We're seeing antibodies again now. They're secreted by plasma cells, also known as plasma B lymphocytes. And then those antibodies have a number of mechanisms they can use to neutralize or destroy that pathogen. And we will get into the details of what those antibodies do separately. All right, that was our main cascade. I think I'll keep recording for a couple of more minutes, but then we're going to go back and we'll pause and we'll start play acting this, and we'll talk about how we're going to gamify that later. So B lymphocytes, those plasma cells, release antibodies. Antibodies are also known as immunoglobulins. Here are the mechanisms that antibodies have to destroy pathogens. The first is my favorite. It's neutralization. So you guys haven't had microbio just yet. So one of the things that you'll learn about viruses, for example, is that the way that they're harmful is that they'll inject their genetic material into your cells and turn your cells into a factory for that virus, which causes more viruses to be released into your system, and in some cases makes the cell explode when it fills up with viruses, which is weird and cool. Neutralization means that they're going to cover the part of the pathogen that's harmful. So in the case of viruses, it would be like covering the part of the virus that can inject its DNA into your cell. So if you want to be slightly obscene about it, but not unreasonably obscene for this, class level, it's like a condom. Yeah, very much like a condom. You slap a condom on that virus, can't inject its DNA into your cells, it can't hurt you. So that's the purpose of neutralization. I like that you arrived there before I even got there. That was good. I was the first person to do that successfully. All right. Neutralization means that we're going to cover the part of the pathogen that's harmful. That way it can't hurt you, even if it's there. We've already seen agglutination before. Agglutination means that we take a whole cell and we clump it with another whole cell. And today I'm specifying the word whole cell because you also have this option over here called precipitation. There's this weird thing that can happen. We can destroy a bacterium, for example, and that bacterium had antigens on it. Those antigens can float freely through our systems after we've destroyed that thing. And if we don't catch them and get rid of them, they can land in our own tissues, and that can cause a form of autoimmunity. That could be one source of why we have autoimmune diseases. We start attacking our thyroid gland because there's some antigen from some pathogen that we already killed that landed on our thyroid gland, and now we're destroying it. So we have this mechanism of precipitation. These antibodies will bind just to the antigen, and it will clump those together so that you can get rid of them. It precipitates it out of solution so that you can pee it out, so that you can excrete it out, so that you can get rid of it so it doesn't land in your tissue and wreak havoc. Yes?
The body generally, like when it has agglutination factors, will it like automatically come with the precipitation? So like we have the cells being broken up, and then like automatically like the antigen should be also.
That's a really good question. I don't know the answer to it. Probably. What's that? Oh yeah, thank you. Somebody want to pass these out? All right, so far we have neutralization, agglutination, precipitation. Now we're going to come to the concept of complement, the complement. What the complement means is that more antibodies are going to help, much like cytokines, basically invite other immunological agents to the party. Last semester, my students convinced me to show them the episode of the Magic School Bus where they where the kid gets sick and they go into his body. It was about 17 minutes of it was completely useless, but there was 3 minutes of it where the antibodies cover the bus, and as a result of the antibodies covering the bus, all of the immune cells started attacking the bus. And that part was accurate, and that part was actually very useful in saying, all of these other cells we talked about, the cytotoxic T-lymphocytes, the helper T-lymphocytes, the memory B-lymphocytes, those are the ones that are also going to be called by antibodies. So antibodies basically will tag things and say, this is the thing you want to destroy, and that brings more immune cells over to that thing, which is really cool. That's called complement activation. The complement is all of those immune cells. And the end product of complement activation is cell lysis. Just like you had with that cytotoxic T lymphocyte, you put that aquaporin in the plasma membrane, that cell dehydrates, it bursts open, you get cell lysis as the outcome for complement activation. which is something that antibodies can do. Okay, that's my stopping point for immunology. I'm going to go ahead and hit pause. I almost forgot to talk about natural killers. Natural killers are going to be a lot like the cytotoxic T-lymphocyte, in that they are going to use perforin molecules as one mechanism to destroy things. The thing that's different about natural killer cells, or NK cells, is that they simply go around and they look for things that are not from our body. That's all they do. So we're all wearing little cellular name tags. Those antigens are little cellular name tags. And all of our cells, all of my Reeves cells, have a little name tag, a little antigen that says, "Hi, I'm Reeves." And a natural killer cell is going around checking all these name tags. And it's going to go over here and say, "What's this cell?" "Oh, it says, 'Hi, I'm Reeves.'" "That's fine, that cell can live." And it's going to go over to this other cell. "Hi, I'm Reeves. This cell can live." And then if it finds a cell that's not a Reeves cell, if it finds a non-self cell, "Hi, I'm Bob," it's going to kill it. It's an assassin. Yeah, it's just going to kill any non-self cell. And here's the really, really cool part about natural killer cells, as if that wasn't enough. They will also kill cancer cells, because cancer cells are mutated. And when that DNA mutates, the name tag potentially can mutate too. So it'll come up to a random cell that's mutated, and it won't say Reeves. It'll say, "Hey, I'm Rives," and it'll kill it, because it's non-self. It's identified as non-self. So we do actually have a mechanism in our body that prevents us from getting cancer. It doesn't work perfectly, obviously. If the natural killer cells were really that powerful, we wouldn't get sick at all ever, because every foreign antigen would immediately be dead. But they are generally out there killing indiscriminately all of the time, which I think is badass. And just a reminder that an antigen is a protein that will be found on a cell or on some type of pathogen, and I'm being very careful with that language, cell versus pathogen. An antigen is a protein that can be identified as self or other, whereas the antibody is that Y-shaped molecule that can help destroy things.